glucose has generated 2 of these. That's all the energy yield that you're getting, ultimately. Another thing you can notice is that many of these reactions are drawn as double arrows, meaning that they're reversible. Like this whole part of the cycle is reversible. But these are not reversible. Citrate synthase, isocitrate dehydrogenase, and alpha ketoglutarate dehydrogenase are not reversible. So they all have large negative delta Gs under cellular conditions, as well as under standard state conditions. Oxaloacetate, by virtue of regulation of many different enzymes in this cycle, always is found in cells at quite low concentrations. And that's important that this be low. If it were not low, this reaction might go in reverse. Okay? The fact that it's low could be a problem for this reaction, citrate synthase, right? You'd like this concentration to be high to help drive that. So the reason that it's not a problem for citrate synthase is because you have this high energy thioester bond in acetyl-CoA. Right? So the low concentration of oxaloacetate helps pull all these reversible reactions toward it. And then the fact that it's low is tolerable because the thermodynamics becomes favorable by virtue of the fact you have high energy bond there. Does that make sense? So these are all the sort of general features that are of importance. Okay? Don't have to worry about remembering the structures of all of these. I will ask questions that will allow you to either deduce the structures or know them. I mean, you know pyruvate, clearly you're going to know acetyl-CoA, but if you get the details of the carbons wrong on some of these other ones, I don't think that's very important, so I wouldn't ask you to questions that would require you to have that memorized. Okay, any questions so far? Okay, so, yeah, sure.
Is the CoA that's made in the first step, is that what goes back to the PDH?
Yes, yes. So this condensation reaction results in splitting off CoA. So now instead of thioester, you've got CoA-SH, the reduced form, which is, of course, is what you need in PDH. So it just goes right back to that. Now, it might go back to some other reaction it needs that also uses CoA. Right? I mean, CoA is used here, too, down on the bottom. So another use for CoA is down here. But yeah. So, you know, this is a very complicated pathway, right? And so you might ask, you know, why do we have to go through all this? Right? So, you know, part of this is that, you know, this is differentiated over, you know, billions of years of evolutionary time, basically. And a lot of these intermediates in the citric acid cycle, these stable compounds, these metabolites, are used as hubs for other parts of metabolism. We'll see a little bit of that as we go through some of the subsequent lectures, that there are connections, especially, for example, when you have something like an alpha keto acid, like alpha ketoglutarate. Imagine what would happen if you took this structure here, and you took this C double bond O, and you got rid of the O, and instead of the O, you had a little H and a little NH3 plus there. Right? What would you have if you did that?
Oh, aspartate.
You would have glutamate.
Or glutamate.
Yeah, you'd have an amino acid. Right? So transaminases are enzymes that convert alpha keto acids to amino acids. Right? So pyruvate is an alpha keto acid. It gets converted to alanine by a transaminase. So that's a key connection between nitrogen and carbon metabolism. And so there are others, right? Then there are connections that are not transaminations, but are other kinds of reactions. So one of the reasons that we can sort of rationalize the TCA cycle is like this, is because it's a hub. And that's very adaptive for the cell to have a central place to feed things into. If the cell is breaking down amino acids, using them for energy, then it's going to find a way to feed them into this cycle. And once they're in this cycle, then they're going to generate the relevant reduced cofactors. We could also say, we also look at it from the point of view of asking, you know, what if we had a situation where we have acetyl-CoA coming in, and so if we split off coenzyme A and just leave ourselves with acetate, you know, shouldn't there be a way to split off CO2 from here? I mean, we've got different ways to decarboxylate. Maybe we could imagine different catalytic power being brought to bear based on carbon-carbon bond. Right? Then we would have CH4 plus CO2. Right? And then maybe we could oxidize this to CO2. Right? In fact, it is possible to oxidize methane to CO2. There's a whole class of organisms that do this. They're called methanotrophs. They're little single-celled bugs that make a living based on oxidizing methane. Methane is their fuel. Basically, they run their cellular metabolism based on methane, which is natural gas, and they put out CO2. For example, this enzyme here is called methane monooxygenase, MMO. Quite an important industrial enzyme, methane monooxygenase. But this enzyme is restricted in its phylogenetic distribution to methanotrophs. We don't have methane monooxygenase. And we don't have the other enzymes in that methane degradation pathway either. So it's not that it's impossible to imagine it getting done metabolically, but it doesn't give you much versatility in your metabolism if you take your product from glycolysis and just break it in half and then oxidize the simplest CH4. Rather, we can take the coenzyme A and incorporate the carbons from coenzyme A and incorporate them into another metabolite, which is oxaloacetate, and make a bigger molecule, citrate, and then split the CO2 off of that. Right? So it's more complicated, obviously, but we get the CO2 eventually from the glucose. It's just that it goes into the metabolic cycle that is connected to all the rest of metabolism, and by virtue of that, it is very adaptive to the cell because it's very efficient for the cell to have such a central hub. Does that make sense? I'm just trying to provide some kind of rationalization for why there's this big thing. So here's this big thing. Okay, so TCA cycle, and we could look at it in terms of what are the types of reactions. We've got a CC bond formation reaction. That's actually a Claisen condensation reaction. It goes through a carbanion. There's a mechanism in the book. We won't be looking at it in detail, but it's interesting. You can follow it if you like. Then there's an isomerization step, so that sort of reaction, a rearrangement kind of reaction, to go from citrate to isocitrate. There's isocitrate here. This is an intermediate that stays on the enzyme, the cisaconate. It's not something that's released and diffuses in the mitochondrial matrix. Okay. We have then two oxidative decarboxylation reactions. Decarboxylation meaning you split off CO2. That's what decarboxylation means. Oxidative meaning that as CO2 is split off, the substrate, the remaining substrate part is also oxidized. Then we have the substrate level phosphorylation to give GTP or ATP. So that's occurring here. And so we're actually able to synthesize it by virtue of the high energy linkage in succinyl-CoA, which is another thioester. If you remember the thio from that table in chapter 13, thioesters and ATP have like the same high energy bond number if you hydrolyze them. They're both at about 30 kilojoules per mole. So if we have an efficient enzyme,